Hi, my name is Shep Bryan. I'm a fourth-year PhD student here at ASU. I'm a part of Presse Lab, which is run by my advisor, Steve Presse. My PhD research is on biophysics and specifically applying Bayesian machine learning techniques to solve complex biophysical problems. I love what I do because it's a blend of biology, physics, data science, probability theory, coding, and a lot more. On some days, I will be in the lab reading up in detail about super small parts of cells and cellular function and thinking about how to model it. Then on the very next day, I will be coding up an algorithm to sample values from a high-dimensional probability space. While those 2 days might seem very unrelated, they are actually very intertwined. Today, I'm going to talk a little bit about one specific project that I have done as a part of Presse Lab. This work was just recently published by Nature Computational Science, so I'm excited to talk about it here today. So let's get started. Today I will talk about my research for our paper titled Diffraction-Limited Molecular Cluster Quantification with Bayesian Non-Parametrics. Our paper was recently published in Nature Computational Science. What we did in this paper is essentially developed an analysis tool aimed at enumerating fluorescent labels in fluorescence microscopy. So, before I can tell you about my research, I will need to introduce fluorescence microscopy and explain why it is important to enumerate fluorescent labels. So then, what is fluorescence microscopy? Well, let me tell you. Basically, when you look at a cell under a microscope, the cell has lots of parts to it. But for the most part, you can't see any of them. You can usually see the overall shape of the cell, and you can make out the nucleus. But for the most part, all of the interesting parts of the cell that you might care about are completely indistinguishable from the parts of the cell that you might not care about. What fluorescence microscopy does is it allows you to see these otherwise invisible parts by labeling them with something that you can see, fluorescent labels. A fluorescent label is exactly what it sounds like. It's just a molecule like every other molecule, except it has a peculiar property where if you shine it with a laser from a laser with a certain laser intensity and wavelength, it will fluoresce, meaning it will emit light of a different color than the light you shine on it. That means that you know what color of light it gives off and you can see the labels. What that means is that, for example, if you want to look at the cytoskeleton inside of a cell, you create tiny labels that you put in the cell that attach to different parts of the cytoskeleton. Then you shine a laser on the cell and you see the actin filaments and the microtubules. So now you have a way to see the cytoskeleton of a cell, which prior to these techniques was completely invisible. So you can see why this is important. So important that a couple Nobel Prizes were given out for developing the field of fluorescence microscopy. Now, there are a lot of different variants of fluorescence microscopy that I won't get into. These include super-resolution fluorescence microscopy, fluorescence correlation spectroscopy, fluorescence in situ hybridization, and many more. What I'm going to do instead is talk about one specific issue with fluorescence microscopy and what we did in my research to address this problem. So we have talked a bit about the advantages of fluorescence microscopy and how it can make something invisible become visible, but fluorescence microscopy does have limitations. In order to talk about these limitations, I'm going to have to switch gears a little bit and talk about a specific type of experiment within biophysics. Lots of things in biology are made up of constituent parts. For example, potassium ion channels are made up of 4 identical subunits. We call a potassium channel an oligomer and the smaller subunits monomers. Now, if you happen to be a scientist studying a protein that you know is an oligomer, one good question to ask is how many monomers make up my protein? At first, you might think that if you don't know the answer, then this is a great problem to solve with fluorescence microscopy, because in theory, all you'd have to do is tag your monomers with fluorescent labels and then count the spots. But if you go ahead and do the experiment and you look under the microscope, you don't see individual spots, one for each monomer. What you instead see is one big blob. The blob you see is due to what's called the diffraction limit of light. And I won't get into it in detail here, but basically all you need to know is that due to the finite size of microscope lenses, any point-sized source of light in a sample under a microscope gets blurred out into a large blob. Studying something big, like the cytoskeleton network, then the size of the blob is small by comparison, so it's not a big issue. But proteins are very small, let's say on the order of 10 nanometers, which means that the blob you see under a microscope can be 10 times larger than the thing you're trying to image. And the blobs from each label completely overlap. So for this reason, it becomes impossible to distinguish subunits of oligomers under a microscope. But hope is not all lost. You still have information about how many fluorescent labels are in your blob, because a blob with many fluorophores in it will be brighter than a blob with few fluorophores. This brings us to what is called counting by photobleaching methods. Counting by photobleaching makes use of the photobleaching of fluorophores, where photobleaching just means that if you shine a laser on a fluorophore, eventually it will turn off. You can think of it like a light bulb, where the fluorophore is shining and then suddenly the fluorophore bursts, which is not exactly what's going on, but it's a great visual. So imagine that we label our monomers, put the protein under a microscope, and then shine our laser on our fluorophores, and we track the brightness of the blob over time. The blob will start off very bright because all the fluorophores are fluorescent. But then as time goes by, the fluorophores burst one by one, and the brightness goes down in a step-like pattern. So now we can record the brightness versus time, count the steps, and then voila, we have the number of monomers in our oligomer. But there are two more problems. Firstly, it turns out that the brightness of the fluorophores follow Poisson statistics, which means that the noise scales with the brightness. This means that for small numbers of fluorophores, the brightness is low, so the noise is low, and thus the brightness in our photobleaching experiment, the steps are very, very clear. But if we try to do the same experiment with lots of fluorophores, the brightness is much larger, so the noise is much higher, and thus the steps are no longer clearly visible by eye. This makes it impossible to count steps when there are, say, more than 20 fluorophores. Secondly, the fluorophores themselves don't have constant brightness. They can fluctuate between brightness levels, and they can go into a dark state where they stop emitting light for a little bit. This dark state transition is called blinking. It's called blinking because the fluorophore will literally blink on and off. Blinking is different than photobleaching because unlike photobleaching, blinking is reversible, meaning that a fluorophore can blink on and off numerous times, but once it photobleaches at the end of the trace, then it no longer turns back on. This means that now if you're looking at your brightness versus time of your fluorophores, you can never be 100% certain that a downward step is due to photobleaching or blinking. So just as a recap, let me explain the problem again from the beginning. Basically, we want to count how many fluorophores we have in a small region of space. We cannot distinguish individual fluorophores from each other when they're close to each other because of the diffraction limit of light. So instead, we blast the fluorophores with a laser and look for step-like drops in the brightness versus time trace where photobleaching occurs. We have to be careful that when we count the steps in our brightness trace, we don't overcount steps when the fluorophores blink on and off. And when there are too many fluorophores, we simply cannot resolve steps by eye. This is where my research comes in. What we did in my lab is we came up with an algorithm that can take in raw photobleaching versus time data and then spit out a best estimate for the number of fluorophores in the blob. So basically, we can enumerate the fluorescent labels even if we can't distinguish the photobleaching steps by eye, and even if the fluorophores are blinking. Now I will explain a little bit about how our algorithm works. In order to address the problem, we are going to use Bayes' theorem. Bayes' theorem is an incredibly useful mathematical equation, and I won't get into detail here, but I will briefly outline the basic parts of it. Basically, in the Bayesian paradigm, we create very detailed models explaining the mechanics of how the data is generated. Then we can invert the equation so that we get a probability distribution for the parameters of the system. The core part of this is that if you treat every parameter in the model as a random variable, which means that each parameter is drawn from a probability distribution, and for every set of random variables, we can calculate the probability of that set of random variables, then we can come up with a best estimate for those variables. So we create a model that explains the data. We start with a probabilistic number of fluorophores. Each fluorophore starts bright, then at each time level, the state of each fluorophore, where the state just means we track whether it's bright or not, or photobleached or not. The state is drawn from a probability distribution conditioned on its state at the previous time level, using what's called a hidden Markov process. The brightness of the blob is just the brightness of a single fluorophore times the number of fluorophores that are bright at that time level. What we record at each time level is a measurement, which is the brightness plus some random noise. We know the value of the measurements because that is simply what we record. So we just simply plug in our observations into this equation, and then we are left with a probability distribution over the number of fluorophores and the states of the fluorophores at each time level. Then all we have to do is maximize this equation with respect to the number of fluorophores to get the highest probability number of fluorophores. Solving this equation isn't easy, which is why it is my PhD work, and I'm leaving out a lot of details that would be tangential to the main point, but all you have to know is that we can solve this equation. So now I will show some results from my method. We have collaborators at Hurton Lab in University of Birmingham, run by Dirk Peter Hurton, who were kind enough to send us data from highly controlled DNA origami fluorophore counting experiments. The data they sent us is raw photobleaching movies of DNA origami constructs with known number of binding sites on them. Using our method, we were able to count the fluorophores in the data sets and get results that are very consistent with the expected ground truth. For example, here I show results on DNA origami with 20 binding sites and 70% labeling efficiency. We expect to find 14 fluorophores on average, and that is very close to what we find. Furthermore, by adding together the brightness of multiple ROIs, we can test how high we can count. We show that using our methods, we can count up to about 100 fluorophores from data where there really isn't any visible brightness steps at all. Originally, I pitched this idea to you as a method of enumerating the number of subunits that make up an oligomer. But there are actually many more applications for this method. For example, proteins like polymerase II are found to often cluster together into large aggregates. We would be able to enumerate the proteins in one of those aggregates using this method. It also turns out that T-cell microvilli have surface proteins that cluster on the microvilli when the T-cell undergoes an immune response. So using our method may provide insight into immune response. So this algorithm will actually help solve a lot of different problems within biophysics. I should point out that we are not the only ones to look at this problem, and there are a lot of good algorithms out there, for example, by our collaborators at Hurton Lab, Dirk Peter Hurton, who focused their method on providing fast and automated subunit counting from large photobleaching experiments. What separates our methods from the rest is that while our method may come with a large computational overhead, our method incorporates a rigorous treatment of blinking and photophysics. This is actually a great improvement because a lot of experiment preparation goes into preparing samples to try to reduce blinking as much as possible. But our algorithm allows one to instead treat the blinking problem from the software side of things. This in turn means that our algorithm can be used on systems where there are no good ways to control blinking, like in vivo experiments. For this reason, I'm excited to see how the algorithm gets used in the future. Lastly, I would like to thank the people who helped make this research possible. I would like to thank my advisor Steve and our co-author Yanni for the mentorship. I would like to thank Hurton Lab at University of Birmingham, and specifically Dirk, Klaus, and Johan for providing me with the excellent data. A big thank you to the NIH and the NSF for funding this. And I would also like to thank Nature Computational Science for publishing our work. I would like to thank you for watching the video. It means a lot that I get to share my research with people. If you liked this video and found it interesting, please feel free to comment on this video or connect with me to learn more. And don't forget to check out our paper, Diffraction-Limited Molecular Cluster Quantification with Bayesian Nonparametrics in Nature Computational Science.